Hey again, everybody, how you doing? Welcome back. We are picking up where we left off from last lecture, and today we're going to be talking about neuromuscular junctions. So if you remember this animation, we spent a little bit of time talking about what's actually going on when we contract our muscles. Then we spent a lot of time looking at the different structures that are going to be involved in the contraction of those sarcomeres, the contractile unit of our muscles, and how these muscles get a little bit shorter. So some of the structures down here we went over last time, and today what we're going to be doing is focusing right here where this neuron is interacting with this muscle cell. Okay, so that's the focus for today. Now, to remind you about a few things and to kind of add in some information onto what we already know about muscles, skeletal muscle will not contract unless it's stimulated by the nerve. Okay, remember that muscle is excitable, and so we need some kind of electrical signal to stimulate it. If there's no nerve connections, then that muscle is paralyzed and won't work, and so we can call that denervation atrophy. So basically when that nerve connection is not working, that muscle isn't used, and that muscle will then shrink. And so if you remember the term atrophy, we went over that way back at the beginning of Unit 2. Now, this information in this diagram, whoa, there's a lot going on here. Okay, so let's digest this for a little bit. You do not need to memorize this entire image, okay? What I wanted to just illustrate with you are some of the things that we've already learned about. And so if we're talking about our spinal nerve coming in here, we've got that posterior branch or that posterior root that's coming back here to that dorsal root ganglion, and that's going to bring information in, our sensory information, back into these posterior or dorsal horns. So here's that spinal cord, and we are kind of integrating a couple of different units here. So remember, we've got those visceral motor outputs, so our autonomic nervous system that's taking branches out to our sympathetic nerves. We have our parasympathetic responses as well. But what we're going to be looking at is that somatic motor output. So coming out of that anterior horn and going out through that anterior or ventral root, and then branching out to different regions of the body and going into those skeletal muscles in our back, or maybe in the muscles of our body or our limbs. Okay, so really that's mostly what I just wanted to show you, is again that integration between the visceral and the somatic motor nervous systems, and where they are and how they're all integrated into these spinal nerves out here. Okay, so lots of information going in and going out and kind of passing all over the place. So there's a term you're going to need to know called a motor unit. And so a motor unit is one nerve fiber and all of the muscle fibers that are innervated by that nerve. So if we look here, for example, we've got our lower motor unit neurons that are coming out of that anterior horn. So we've got our synapse right back in here. And if we look at the blue one and we follow the blue one out, so we're coming out through that anterior root into our spinal nerve, and that motor neuron, so we can call it motor neuron 1, for example here, is going to branch, and it branches out to a muscle cell or a muscle fiber there, and a muscle fiber here, and a muscle fiber here, and another muscle fiber over here. So this blue neuron is going to innervate these 4 blue muscle fibers. And so if there's an action potential sent down a neuron, which we know that's how these work, it's going to stimulate and excite these 4 muscle fibers. Well, on the other hand, if we look at our red motor neuron, or motor neuron 2, same pathway, and it's coming out to the same muscle. Remember, these little branches of this nerve are penetrating through all that connective tissue of that paramycium and that endomycium right down into each of those muscle cells. Now, with that red motor neuron coming down, you can see that it is innervating these red muscle fibers, these 4 red muscle fibers. So if motor neuron 1 fires, only these blue muscle fibers fire. If motor neuron 2 fires, only these 4 nerve fibers will be contracted. But if both motor neurons fire, then we actually have 8 muscle fibers that are contracting. And so these motor units then allow us to have this dexterity, right? So we can fire a few neurons, or we can fire a lot of neurons. And so kind of the average is about 200 muscle fibers for one of these motor neurons. Now, if we have like really fine motor skills, we might only fire 3 muscle fibers. But with really big muscles, we might stimulate thousands of muscle fibers to have really big contractile forces. So let's talk more about that neuromuscular junction. Okay, remember, a synapse is where a nerve fiber is meeting a target cell. And so in our case, neuromuscular junction, neuron and muscle, is when a target cell is a muscle fiber. Okay, so those synapses can be between other neurons or other organs. And in this case, when it's a muscle fiber, we call it the neuromuscular junction. Each of those branches then are forming a separate synapse with that muscle fiber, as we just saw with that previous image of those motor neurons. And one fiber then actually will stimulate a muscle fiber at several points along its length, okay? And so when you have that branch coming out, it's not just going to one spot on that muscle fiber, but remember, those muscle fibers can be really long, and so we need to stimulate at different locations along that muscle fiber. So I think this is just a really awesome image to kind of give you a perspective of what this neuromuscular junction looks like. So you can see these motor nerve fibers coming down, or these axons that are coming down and synapsing with these individual fibers. So we've got one coming in here, we've got one coming over to this muscle fiber. Looks like we've probably got one that's coming out and going over to this muscle fiber, right? So just a pretty cool image of this junction. Now, what I would recommend you doing is pausing the video here and really thinking about Unit 3, Unit 4, and our last lecture, 19, over the characteristics of muscles. And really test your knowledge at this point. You know, how much do you know? What do you not know? What are things that are kind of foggy? So take what you know and apply it to this slide, this neuromuscular junction slide. We can also call this a motor end plate. And on the next slide, I've got a bunch of questions. So be looking at this, all of the structures here. What do they do? How are they involved? What is their functionality? So what are you familiar with? What definitions do you remember? Can you write them out? How's this similar to Unit 3? How does Unit 4 tie in here? And can you make some predictions about what's going to happen at this neuromuscular junction? Given what we know about Unit 3, can you make some predictions about Unit 5? And then try to connect everything you know about neurons up until this point. Okay, so we'll see you back here in about 10 or 15 minutes, and then we'll move on and figure out how all this works. All right, welcome back. How did it go? Well, let's look at how all of this works. Hopefully you were able to really think about everything, really dig deep into those folds of your brain, those gyri, really integrate that information that you've been sticking into your long-term memories, and be able to draw it out and make sense of it. So a couple of other points that I wanted to review with you. So muscle fibers and our neurons are both excitable, okay? That's really important. When it's unstimulated, we have what's called a resting membrane potential, right? Or a resting cell. So that plasma membrane, though, remember, is electrically polarized, so it's charged, okay? So we're going to see the same thing with our muscle fibers. We have a higher concentration of sodium outside of the cell, and we have a higher concentration of potassium inside of the cell, okay? And remember, we have our sodium-potassium pump that is using ATP to drive that and move these ions up their concentration gradients, and the higher concentration is going to create that potential, right? That current when those ions move through those channels. We also have anions inside of the cell, and so overall the inside of that plasma membrane is negatively charged. All right, so keep that in mind. So that then leads us into our electrical potential, or our voltage, basically our difference in charge from the outside to the inside, and with our resting membrane potential. So at rest, in a muscle cell, our voltage is about negative 90 millivolts. And again, that sodium-potassium pump is helping maintain that. Again, we have some of those leak channels that are also playing a role in that resting membrane potential. Remember, with our neurons, we talked about negative 70. So here we're going to see a little bit of differences in some of these voltages. Again, here's our resting membrane potential for our neurons at negative 70. We get excited, excitatory signals coming in. We reach a threshold and boom, we fire. We open all those sodium, voltage-gated sodium channels. Our potassium channels are slowly opening. Then our sodium channels are closed and we repolarize. And so we have our absolute refractory period here, which is helping move this signal in one direction. We have our hyperpolarization and our relative refractory period, which is again helping us move that action potential in one direction. And then our return back to resting membrane potential. So what we're going to look at now are basically 4 different phases that kind of play a role in our contraction and relaxation. So our first is excitation. So basically our nerve action potentials are going to cause or stimulate action potentials in our muscles. Okay, so that should be expected, right? We learned about that with our neurons stimulating an action potential in another neuron if it's excitatory. Our second phase is the excitation contraction coupling. Okay, so here we're exciting and we're going to start contracting. So we're basically taking those action potentials that were a result of that communication from the nerve that are traveling down that plasma membrane of the muscle fiber, or the sarcolemma, and that's going to activate our myofilaments. So we have to take our action potentials, so this electrical signal, and make these myofilaments do something, okay? And then we have our contraction phase. So now our muscle fiber is going to shorten and get shorter. So we're going to bring those Z discs of the sarcomere closer together. Remember, those myofilaments are not getting shorter. They're just moving over the top of each other and overlapping a little bit more. And then we have our relaxation state, so that muscle fiber is returning back to its resting length. Okay, so this is what we're going to go over during this lecture. Now, this should hopefully look familiar, because this was what you hopefully spent about 10 minutes or so going over and practicing your terminology. So basically what's happening here is, if you remember, here's our nerve signal, our action potential that's passing down our axon, and it's coming up to our neuromuscular junction here. So we can see that we've got our muscle fiber down here. We can see this alluding to the presence of those myofibrils filling up that sarcoplasm, and here's our sarcolemma, that plasma membrane of that muscle cell. Okay. Now remember, when that nerve signal comes down that plasma membrane, it's stimulating calcium, and calcium moves into our synaptic knobs right down here, and by calcium moving into the synaptic knobs, it's stimulating the release of, hey, our good friend acetylcholine. And so those acetylcholine molecules or neurotransmitters are being released from those synaptic vesicles, and it's being released into our synaptic cleft. So over here in image 2, you can see, or step 2, you can see that acetylcholine being released. And if you zoom in right in here, hey, look what's there, acetylcholine receptors. So 2 acetylcholine molecules are going to bind to those receptors. Now, if you're thinking about this, hopefully you're already a couple steps ahead and you're saying, well, excitation. So if acetylcholine is being released, well, I know there could be either a muscarinic receptor or a nicotinic receptor. And hopefully you're saying to yourself, well, I want to excite this muscle. Therefore, if I'm going to excite it, I need to get closer to threshold. Therefore, I need to allow positive ions into the cell. And by doing that, it must be a nicotinic receptor that is present on the sarcolemma here. Right? If you remember, the muscarinic was that second messenger, that G-coupled messenger system that then opened potassium channels and had that inhibitory effect. So if we move to the next slide, oh, bingo, here's our nicotinic receptor protein. So good. Hopefully you were thinking that and you were able to work yourself through the logic and say, great, sodium's coming in, a little bit of potassium out, but not as much as sodium is coming in. And so that is going to trigger an excitation, right? It's going to increase our increase the charge from negative 90 millivolts to positive 75 millivolts. So different numbers here. So we have that action potential, or we have that quick voltage shift, and we call that an end plate potential. So remember, we called that neuromuscular junction, that motor end plate, and so here we can call it an end plate potential. Now, if we look here again is that plasma membrane of that knob. We've bound our acetylcholine here, and so that voltage change is going to open our potassium and our sodium channels. And that is going to then stimulate an action potential that's going to pass down that plasma membrane in one direction, right? Because we still have those refractory periods following those action potentials. So it's going to spread over that muscle surface thanks to our sodium rushing in. Now what we want to do is couple that excitation with contraction. So we want to get that action potential, and we want to stimulate these myofibrils, or those myofilaments in the myofibrils. Okay, so you can see here these little arrows. Well, what this is representing is our action potential, and so it's passing down and across our sarcolemma. But if you remember, we said those T-tubules are infoldings of that plasma membrane, and so they are that phospholipid bilayer. So that action potential actually passes down and into this T-tubule that's now passing down deeper into that cell. When it does, it actually is opening calcium channels in that sarcoplasmic reticulum. So over on the left, we can see a storehouse of calcium. On the right, we see that calcium being released into the cytosol, okay? And so that calcium now is all spread out throughout that cytosol, and it is surrounding our myofibrils and our myofilaments. And so hopefully now you're thinking about, well, okay, calcium is really important, so how is this going to do anything? All right, well, if you remember our thin filaments with actin, we said we had these binding sites on those little subunits, but they're covered up with the door, which is tropomyosin, and they're locked with troponin, but troponin has the little calcium binding sites, and so the calcium then is our key that's going to fit into that lock. And so calcium is going to bind to troponin, and that troponin-tropomyosin complex has a shape change, and that's going to then open up these pathways, or these binding sites, these active sites, and now we can bind to our myosin heads. All right, great. So calcium is the key. It binds to troponin. Our troponin-tropomyosin change shape. We open and the active sites, and now we can have binding between the active sites on our actin and our myosin heads. So Now what we want to look at is our contraction. So here we're kind of coupling our excitation with our contraction. The excitation stimulated calcium release. Our calcium release has now enabled the ability for these proteins to contract. Now what's happening with contraction? Okay, so what we're looking at is then the binding of, well, ATP is hydrolyzed to ADP and a phosphate. And so this is going to activate this myosin head. So it kind of cocks this head back, right? And then what we see happening is as it's stuck in this cocking position, it binds to that active site on that actin subunit, and we call this a crossbridge. Okay, so this is called a crossbridge. That's a really important term to be familiar with. Then what happens is that ADP and phosphate are released, and that myosin head cocks back, or pulls back, I should say, in what's called the power stroke. So it pulls on this thin filament, and you can see this thin filament shifts from here over to the right. And so that's our power stroke. Then what we see happening is an ATP binds, and it breaks that crossbridge. So now, if we go backwards, that ATP is hydrolyzed, that myosin head cocks into its extended position, it binds to the active site, we release ADP and phosphate, we have our power stroke, and then ATP breaks the crossbridge. So it's going to bind with another active site, and then another active site, and another active site. And so each head will have about 5 power strokes in a second. So, if you think about like the sheer numbers of this, like thousands and thousands and thousands of these little myosin heads binding and utilizing thousands and thousands of ATP molecules, right? This is why we have lots of mitochondria to just continuously crank out ATP to feed these things, essentially, right? So that we can have these sustained contractions and prolonged muscular activity. All right, now when we talk about sort of relaxation here, that nerve stimulation stops. And if that nerve stimulation stops, well, then no calcium is coming in. If no calcium is coming in, no acetylcholine is being released, and any acetylcholine that is in that synaptic cleft, or in that space there, is being broken down by acetylcholinesterase, and so we lose the binding sites to those receptors, and therefore those receptors close, and now we don't have sodium coming into that nerve muscle cell. Okay, so we stop that stimulation, and that muscle can then return. Okay, and so when we talk about that continued relaxation, well, inside of the cell, if there's no more stimulation, we're seeing that calcium is getting pumped back into the sarcoplasmic reticulum through active transport. So again, we're using ATP to pump that calcium into our sarcoplasmic reticulum and clearing out the calcium that's available. And so when we are losing that calcium, there is no more key, and so we pull basically the key out of our lock of that troponin-tropomyosin complex, and that tropomyosin and troponin complex then cover up those binding sites or those active sites on those subunits, and those myosin heads can no longer bind to that actin, and that muscle then returns to its original length. And so part of that is from that titin, and part of that is also from antagonistic muscles that may contract and stretch the opposing muscle. Now, when we pass on, we tend to see something called rigor mortis. And so this is the hardening of muscles that generally happens within a few hours after death. So what's going on with rigor mortis? Here you can see a couple of images of some people that are sort of in this state where their muscles are just super hard and rigid. So as our sarcoplasmic reticulum and sarcolemma are breaking down and releasing calcium, it's kind of activating that cross-bridging. Okay, so we get this flood of calcium. It opens up those active sites. The myosin heads bind, and we get these cross bridges. Well, it requires ATP to break those cross bridges. And so ATP then is no longer produced after we die. And so we use up any ATP, and these cross bridges then sort of remain contracted until these myofilaments start to break down. Okay, so this kind of peaks about 12 hours after death, and then generally after about 2 days kind of declines over time. Now, a couple of connections with some toxins. So they are going to interfere with that synaptic functioning, so between the neuron and the muscle. And so spastic paralysis is a type of paralysis where we have these continual contraction of the muscles. Okay, so some chemicals, pesticides, will inhibit cholinesterase or acetylcholinesterase. And so if you remember, we talked about nerve gas earlier in Unit 3. And so they will bind or inhibit acetylcholinesterase, and they prevent it from breaking down acetylcholine, and therefore if you don't break down acetylcholine, you have more binding to those nicotinic receptors, more sodium, and you have lots of these sustained contractions. Tetanus then is a type of spastic paralysis, and it's caused by a toxin that comes from the bacteria Clostridium tetani. And so if you remember our neurotransmitter glycine was in the spinal cord, and it was inhibitory. It stopped motor neurons from producing unwanted muscle contractions. So if this toxin then that's released by this bacteria blocks glycine, and so if you're blocking glycine, then you're not inhibiting unwanted muscle contractions, and so now you're experiencing unwanted muscle contractions, which causes this spastic paralysis. Now, on the other hand, flaccid paralysis is where your muscles can't be contracted. So curare, you've probably heard about curare. It's used by South American natives. They'll put this on blowgun darts, shoot it at the prey that they're hunting, and what it does is it actually competes with acetylcholine, and so it binds those receptor sites, but it doesn't actually stimulate the muscles to contract. So acetylcholine can't cause excitation and allow those muscles to contract because curare is bound to those binding sites and keeps them from being stimulated. And then botulism. Botulism is food poisoning by a neuromuscular toxin called botulinum, which is produced by Clostridium botulinum. And again, this blocks the release of acetylcholine. So again, we're not going to have acetylcholine that's going to bind to those nicotinic receptors, which is going to open up sodium channels and then create excitation. So Botox comes from this toxin, and this is used in cosmetic injections to reduce wrinkles. So we have made it, okay? We have connected our neurons to our muscles, and we've learned about how all of this works. So my suggestion to you, as always, I'm sure you didn't see this coming, is to spend about 10 or 15 minutes going over this animation and walk yourself through everything that's going on and all of the structures and how all of this works. Practice this over and over again to help paint the bigger picture. As always, here's some review and reinforce questions for you at the end of the chapter or at the end of the lecture to practice and reinforce that content. And as always, let's build on Unit 3, Unit 4, and Unit 5. And so as we're going into the winter, although it's a La Nina year, it may not be that great of a winter, and so we'll see what is to be expected. It is 2020. Who knows what will happen in 2020? There is still time left. Sorry about that. So you're walking around, you slip on some massive piles of snow and ice that were not shoveled off of the sidewalk as you're wandering around campus because we are in COVID isolation. As you slip, whoa, you get some sensory information sent down the vestibulocochlear nerve. Remember, our inner ear, the semicircular ducts, and our vestibule are sending in information about balance. And so we're sending that back to your brain. You should practice and revisit all that information. What structures are involved? How is that moving along? Take what you learned in lab and tie it all together. So we send an action potential down our upper motor neuron through that vestibulospinal tract. Hey, remember that one? Let's practice that. And that goes to our lower motor neuron. So starting with an EPSP at the dendrites of that lower motor neuron, what is going on? Take us all the way from this anterior horn out to our muscles, and how does that integrate and make us catch ourselves and keep our posture? Okay, so practice this entire pathway. There's a lot that's involved. So we'll see you next week. Hope you have some time to kind of process all of this, and we will continue on next week with more information about muscles. Thanks. Have a great day.